Hello and welcome to the EPG Pathshala program in Botany. I am Dr. Hemlata Kotkar, currently working as an assistant professor at the Department of Botany, Savitribai Phule, Pune University. I have completed my Masters and PhD in biotechnology. Later, I joined the CSIR National Chemical Laboratory as a postdoctoral fellow. I have also got an opportunity to work at the Max Planck Institute for Chemical Ecology at Jena, Germany, twice as a Max Planck India visiting fellow. Under this module in botany, I would be talking on plant physiology. The subject would be plant mineral nutrition. Plant growth and productivity is directly affected by availability of water and plant nutrients from the soil. Plant biomass accumulation and yield directly depends upon the availability of plant nutrients. These nutrients are broadly classified as macronutrients, micronutrients, and secondary or beneficial nutrients. In this module today, I will be focusing on iron and sulfur. In this module, we will focus on soil availability, uptake and transport in plants, functions and deficiency symptoms of iron and sulfur. Plant growth and productivity is mainly affected by water and mineral nutrient availability in the soil. Amongst these, macronutrients are required in larger quantities. They include nitrogen, phosphorus, and potassium. Micronutrients are required in smaller quantities and are involved in all metabolic and cellular functions. Plants differ in their need for micronutrients. They include iron, copper, molybdenum, boron, manganese, zinc, chlorine, and cobalt. These categories are based mainly on requirements by the plant. Concentration of macronutrients required as per dry matter is more than 1 mg per gram and is less than 1 mg per gram for micronutrients. In this context, it is important to note that secondary nutrients are those which are required by plants in lesser quantity than macronutrients and in more quantity than micronutrients. Secondary nutrients include sulfur, calcium, and magnesium. Use and efficiency of macronutrient absorption is dependent on availability of micronutrients. Let us begin with soil availability of iron that is considered as a micronutrient. Iron, being an essential nutrient, is required by plants in the largest amounts. It is an abundant element in the earth's crust, but the concentration of soluble iron in the soil is low. It may be taken up by plants as the ferric, that is Fe3 form, or ferrous, that is Fe2 iron. Fe2 form is more common due to its high solubility, which can be absorbed directly by some plants. Fe3 has very low solubility in water that is affected by both pH and oxygen. However, under aerobic conditions, when soil pH increases above 6, Fe2 readily oxidizes to Fe3. The soil concentration of inorganic iron under such conditions is less than 10 raised to the power 16 molar, which is lower than required, that is 10 raised to the power 8 molar for plant growth. Most soluble Fe in soil solution is chelated by organic and inorganic ligands. As the concentration of soluble iron chelates remains very low, plants use several mechanisms to acquire iron from the soil for growth requirements. In contrast, in acidic waterlogged soils, Fe2 can be toxic to plants. Plasma membrane localized components include a plasma membrane H+ATPase that acidifies the rhizosphere and increases iron solubility. In Arabidopsis, the gene AHA2 codes for H+ATPase. Reduction of iron at the root surface is controlled by NADPH dependent ferric reductase FRO2. FRO2 reduces Fe3 chelates in the rhizosphere, releasing Fe2. This enzyme is a member of flavocytochromes that transports electrons across membranes. The high affinity transporter called as IRT1 or the iron regulated transporter 1 transports Fe2 across the plasma membrane. IRT1 is a member of the ZIP or the ZRT-IRT related protein family present at the plasma membrane of root epidermal cells. The figure shows protein structures of the members of ZIP family of transporters. 8 membrane spanning domains and a histidine rich cytosolic loop that functions in the metal binding are seen. In the grass family, phytosiderophores are released from the roots that chelate and solubilize Fe3, making it available for uptake, called as the strategy 2 mechanism. Graminaceous species are less susceptible to iron deficiency than dicots. Grasses differ in the response as they do not increase H+ excretion or ferric chelate reductase activity. They release non-protein amino acids called phytosiderophores from their roots that chelate ferric iron and solubilize it. After Fe3 is chelated by a phytosiderophore in the soil, the complex is transported by proton-coupled transporters into the root cells. These transporters belong to the oligopeptide transporter family called as OPT. Transport of iron from the root to the shoot takes place via xylem as well as the phloem. In the xylem, iron ligand complex diffuses through symplast to the stele and is released into the apoplast. Iron is bound by citrate and other compounds in the xylem to be released in the transpiration stream. A mutant called FRD3 or the ferric reductase defective 3 reveals the role of citrate in the movement of iron to the shoots of Arabidopsis. Iron deficiency responses were upregulated in the mutant plants but show typical symptoms under normal conditions. The gene encodes a transport protein from the multi-drug and toxic compound extrusion abbreviated as M-18 family. Iron is involved in photosynthesis, mitochondrial respiration, nitrogen assimilation, biosynthesis of hormones such as ethylene, gibberellic acid, jasmonic acid, production and scavenging of reactive oxygen species, osmoprotection, and pathogen defense. The uptake of iron is related to two important functions in the plant. First, it is a part of the catalytic group for many redox enzymes, and second, it is required for the synthesis of chlorophyll. Up to 80% of iron is found in the chloroplast. Although a direct role in the chlorophyll synthesis is not attributed to, it is related to synthesis of electron transport proteins. Redox enzymes such as the heme-containing cytochromes and non-heme iron sulfur proteins are involved in photosynthesis. Iron is also a part of oxidases such as catalase and peroxidase. The well-known hemoproteins are the photosynthetic and respiratory cytochromes involved in electron transfer and the globins that bind oxygen. Iron deficiency in the soil is rare, but it can be unavailable for absorption if the soil pH is not between 5 and 6.5. A common problem associated with iron deficiency is excessive alkalinity of the soil, that is, the pH is above 6.5. However, iron deficiency can also develop if the soil is too waterlogged or has been fertilized excessively. In general, iron deficiency is a plant disorder also known as lime-induced chlorosis. Chlorosis can be explained as yellowing of leaf tissue due to lack of chlorophyll. Deficiency symptoms of iron include the leaves turning yellow or brown in the margins between the veins, which may remain green. Young leaves may appear to be bleached. Overall, chlorosis might lead to poor quality and quantity of fruits. Let us now shift to sulfur. Sulfur is an essential element of plant growth, development, and response to environmental changes. It is required for biosynthesis of proteins, is involved in nitrogen fixation in leguminous plants, and also helps in seed production. Sulfur adds color, a different smell and flavor to various plants like garlic, brassica crops, and onions. The nutrient also forms a significant part of proteins and amino acids. Sulfur is crucial for ensuring the protein content of leguminous plants. The biogeochemical sulfur cycle is the interconversion of oxidized and reduced sulfur species on Earth. Soils contain both inorganic and organic forms of sulfur. Amongst the several other forms of sulfur found in the soils, iron sulfides and elemental sulfur are also important. In aerobic forms, sulfur is mainly present as sulfate, the form by which plants take up most sulfur from the soil. The reducing environment created by flooding can give rise to sulfides such as FeS, FeS2 and H2S. In addition to soil sulfur, plants can also extract it from the atmosphere where it occurs as sulfate and H2S. Numerous organisms are capable of oxidizing sulfides or decomposing organic sulfur compounds. Hence, sulfur deficiency is not a common problem. Sulfate is highly mobile and is rapidly transported through the xylem to shoot tissues. Reduction of sulfate takes place in plastids where it is assimilated in cysteine. Cysteine can be directly incorporated into proteins or converted into methionine as shown by the number 1 in the figure. Cysteine contains SH, that is, the thiol groups essential for protein functioning through the formation and disruption of sulfur bridges depicted by 2 in the figure. Reduced sulfur shown by 3 is mainly distributed around the plant as the tripeptide glutathione. During many stresses, glutathione-based phytochelatins or PCs are synthesized which detoxify metals and metalloids. There are 3 main types of sulfur transporters. The first one are the ATP dependent ABC type transporters. The second ones are the sodium proton sulfate symporters, and the third ones are the sulfate anion antiporters. In higher plants, only the proton sulters are present. SULTRs are the sulfur transporters. Plants' sulfur transporters or the sulters are integrated into the membranes by 12 transmembrane regions and contain a STAS domain. The STAS domain is important for the correct incorporation into the membrane, activity and interaction with other proteins. Sulfate uptake is induced when sulfate is limiting and is repressed in the presence of reduced sulfur. The figure shows multiphasic rate of sulfur uptake by barley roots when incubated with a range of sulfate concentrations. At low nitrite levels and the availability of carbohydrates, sulfate uptake is repressed. Regulation of sulfate uptake is well correlated with regulation of mRNA levels of the transporters. Sulfate starvation results in increased sulfate uptake and induces transcript levels of Arabidopsis sulfur genes. The transcript levels of sulfurs are rapidly reduced when sulfate is resupplied to sulfur-starved plants. Sulfur deficiency is not a common problem because there are numerous microorganisms capable of oxidizing sulfides or decomposing organic sulfur compounds. Sulfur deficiency, similar to nitrogen, results in the generalized chlorosis of the leaf. The tissues surrounding the vascular bundles also show chlorosis. This is an effect due to reduced protein synthesis rather than a direct impairment of chlorophyll synthesis. However, chlorophyll is stabilized by binding to the protein in the chloroplast membranes. The ability to form stable chlorophyll protein complexes is also affected due to impaired protein synthesis. Sulfur is not readily mobilized in most species, and the symptoms tend to occur initially in the younger leaves. Following are the deficiency symptoms of sulfur observed in many plant species. The first one, it results in yellowing of the whole plant. Younger leaves become yellowish white, that is, they develop chlorosis and their tips become necrotic. Overall plant development and growth is stunted. Sulfur causes higher mortality after transplantation. Importantly, sulfur deficiency adversely affects the baking quality of wheat and yield of oilseed brassica species. It is also associated with increased susceptibility of crops to insect and fungal attack. Sulfur plays some major roles in plants. It is found in some amino acids that serve as the building block of proteins. Sulfur is essential for chlorophyll formation. It is a major constituent of one of the enzymes required for formation of chlorophyll molecule. Sulfur is required in the synthesis of oils, especially in oilseed crops. It is active in the metabolism of nitrogen. Sulfur is mostly found in reduced form in the amino acids cysteine and methionine. In cysteine, sulfur appears in the sulfhydryl or the SH-thiol groups that can undergo reversible oxidation and hence the formation of a covalent sulfur bond if a second sulfhydryl group is present. The production of glutathione serves as a mobile carrier of reduced sulfur. Another role of inorganic sulfur is in sulfolipids. These are normally found in a small proportion in chloroplast thylakoids. Sulfur toxicity is rare but can occur in saline soils with high levels of sulfates. Soil availability, uptake and transport, functions and deficiency symptoms of iron, which is an important essential micronutrient, and sulfur, which is a beneficial nutrient, have been discussed. The improved understanding of specific amounts required and the intensity of interaction between different species and forms of nutrients present a major challenge in knowing the dynamics of the biogeochemical cycle in soil. Such information practically enables identification of methods against deficiency. In a broad scenario, it would help to restrict the onset of deficiency symptoms and finally crop yield limitations as a whole. In this module, we have learned about soil availability, uptake and transport, functions and deficiency symptoms of iron and sulfur. Both the elements are very essential to the plant for growth and productivity. Different species of iron exist which have an intrinsic behavior in the plant. This coordinated behavior between different elements is necessary in the agricultural scenario for productivity and yield as a whole. Study of these two elements is therefore necessary to learn more about the plant, its yield and productivity in an agricultural scenario as a whole. Thank you for patient hearing.